I would like to present a report on analysis of DNA and RNA sequence using next generation sequencing, and the results are analyzed using bioinformatics tools. The project was done by myself, Dilna, Damodaran, and Sreejith, Balakrishnan. In this slide, I would like to summarize the importance of next generation sequencing, which is a massive parallel sequencing technology known nowadays, which has revolutionized biological science. The next generation sequencing are used are widely used in medical research nowadays, mainly to understand the tumor DNA, to understand the targeted exomes, to study the targeted exomes, to create a genomic library for microbial profiling, and for detecting the mutations. So the sequence obtained through next generation sequencing can help us to understand somatic mutations, polymorphisms, assembled genomes, community characterization, and structural variants. So next, move on to the DNA sequencing results analysis after next generation sequence. The present slide explains the application of next generation sequencing and how the data is processed using them. The sequence which is obtained through next generation sequencing are analyzed using various bioinformatics tools. These bioinformatics tools help us to understand the somatic mutations, the polymorphisms, the assembled genome studies, then the variation in the cell structures or the genomic construct, etc. This particular slide explains the pipeline of DNA sequencing, DNA sequence after the next obtained through next generation sequencing. The raw data analysis, which is the result of next generation sequencing, is in the form of FASTQ. The FASTQs are read to analyze its quality using you can use two bioinformatic tools, namely Bowtie and BWA. So any of these tools can be used to analyze the quality of FASTQ sequences. Once the quality is comparable, or once you have a good quality, or in case if you don't get a good quality sequence, you can again modify your sequence to improve the quality. So once the quality is satisfied, then you can go ahead and move to the next steps, which is aligning them to a reference genome. This reference genome can be obtained through from the genomic library. In case of human beings or Homo sapiens, such reference genomes are obtained from human genome projects. The sequence, the results obtained using bioinformatics tools like Bowtie and BWA are in .sam files. These .sam files can be converted to the binary formats using SAM tools. Once they are in the binary format, it can be further formatted it can be sorted and then indexed. Then by using another bioinformatics tool called Freebies, these sorted indexed BAM files can be converted to generate or can be used to generate a VCF file. So let me give you a small overview of VCF file. VCF file gives us key informations like the format, date, the software used, the human genome used for the referencing, Then the SNPs, that is single nucleotide polymorphisms, are explained are clearly mentioned in the VCF files. The VCF file also you can also find the genotype, the read depth and the frequency of genome under the study. This is an overview slide which explains the accession number of the DNA sequence used for the study, and which is SRR7143692. And standard protocol is followed to obtain this sequence. The present slide explains about the details of the study during which the DNA sequencing was done. So the study was about the energetics and physiological impact of cohesion extrusion in mammalian cells. So this is an overview of FASTQ files obtained for the DNA sequence of accession number SRR7143692. This is the quality check results of the DNA sequence or the FASTQ files using a bioinformatics tool FastQC. The FastQC report of sequence alignment are as follows, and as the result is which clearly explains that a good quality sequence was obtained. This slide explains the results which are obtained after the alignment of of the studied DNA sequence with the reference sequence. Using bioinformatic tools BWA or Bowtie, here I have used BWA to analyze the alignment, and the SAM files obtained are then converted to BAM files using the SAM tools. The BAM file is the .bam files are then processed after marking and sorting. This slide shows the IGV view of .fam files aligned with chromosome 20 of from the library, that is of the Human Genome Project HG38. This is the headers of .pcf files obtained after analysis, which explains about the format, date, the source, the reference sequence, and various IDs used for the studies. Next, we will move on to the next generation sequence, the RNA sequence obtained after next generation sequencing and its analysis using bioinformatics tools. This is an overview slide which explains the pipeline for RNA sequencing. So which clearly explains that initially we have the sample preparation where genomic RNA is extracted, and then followed by the library construction, then followed by sequencing using Illumina. The raw reads are in the FASTQ format similar to the DNAs. The quality of these FASTQ formats are understand using FastQC. Using a bioinformatics tool called FastQC. These are the results. The resulted document, that is the resulted file, which is in the .sam format, are then aligned using BWA. After the alignment, the faster sequence which are obtained after the QC are then aligned using a reference genome. Here for my study I have used chromosome 6. So after the alignment, the alignment was done using the tool BWA, the result of the alignment was in the form of .sam files, and these .sam files are then processed to convert them to .bam files, and which are used for further analysis. The RNA sequence obtained for the study are from a research on the psychiatric disorders. The concentration, the RNA sequence are of HLA-DRB1. This HLA-DRB1 are present on T4 cells, and which are the major immune responding cells. So the RNA sequencing preparation methods are as follows. Once the total RNA was extracted, using a standard extraction protocol, a hybridization was done to obtain a cDNA. The cDNA using the cDNA obtained through Illumina high sequence 2500 or 2500, which clearly explains what was the input, which is the whole RNA sequence, and the results of Illumina or after next generation sequencing was a paired cDNA. And the basic standard protocol followed are as follows. So the accession number of the sequence under study is SRR5252865, and the reference chromosome used for the study is chromosome 6, which has an NCBI accession number NC_000006.12. The resulted files of the study are as follows. This is the QC results of the sequence SRR5252865. It shows not a very high quality but moderate quality to be. So hence we further carried on with the study. The alignment result of the study sequence with the reference sequence chromosome 6 was viewed or for a better understanding. We viewed it using IGV, and the alignment was clearly shown. A differential genome expression equation was carried out for this particular study, and the box plot obtained as a result is as follows. This slide explains shows the p-value of the differential genome equation calculations, and which clearly says there was a significant least p-value was obtained for the ID 4035106. Thank you.